Oh, thanks. My first words, they go to the organizers of this meeting for the kind invitation to be here today and share with you this topic. The title you can see on my slide. And also, I'd like to thanks all of you online attending this meeting and in particular attending my talk. My pleasure to be here. It's an honor to me. So today I selected this topic to share with you. So I will presenting to you a combination of radiofrequency therapy and nanomaterials. So actually it's a nano composite. And the point that I want to emphasize here is the synchronism between this radiofrequency therapy and the use of nanocomposites that I will describe. So there is a huge synergy between these two approaches. And this is the topic I want to share with you. Here is my agenda for today. In the analysis of the data, I will show you a mathematical model. And then in my first and second slide, I would like to emphasize the use of mathematical modeling to analyze cell and actually today it's only cell viability data. But also I have been working on in vivo experiments like biodistribution. And then very surprisingly, I would guess you will be surprised to see how little is a mathematical model has been used to analyze a bioassays. So it's the second point in my talk. And then I will switch to present you the radiofrequency very shortly, and also the nanocomposite based on graphene oxide. So, and then I will switch to the in vitro experimental design, combining radiofrequency and the use of this nanocomposite. And finally, I will analyze the data using the Hill model, and I will end my talk with some conclusions. So here is a very interesting aspect about in vitro data. If you do a search on Web of Science, for instance, about in vitro data, you will find a huge amount of papers, close to 3 million papers in vitro assays. However, only a very small fraction, less than 1%, includes any mathematical analysis in the data. And that is, of course, a big challenging to jump from a qualitative analysis to quantitative analysis. And the reasons of this, let's say, very small fraction of quantitative analysis is perhaps quite obvious. So most of the people working on in vitro assays, the background, the math background, perhaps it's a blocking aspect to use mathematical modeling. The second point is, as you have a mathematical model described by a, let's say, one equation, you have to use that equation to fit the experimental data. And then you might need a protocol to perform this curve fitting. However, we have a very effective approach to do this, which is called the least square feet. And least square feet is around for almost 3 centuries. So despite of this, we have a very small fraction. And the good thing about this novelty, perhaps to you, is that in the last 2 3 decades, the number of papers using mathematical model is increasing. This is a very good hope for the future. Although the total number of papers, including mathematical model, is still very small. I would say less than 1%. And let me show you the typical in vitro presentation data you may find these days in a paper. So I just took this paper here from last year. And in this paper, they are analyzing the one chemotherapeutic agent, which is echinacin. And to perform the in vitro assay, they use two different cell lines, both neoplastic cell lines, the T24 and the EJ. So these two cell lines were challenged by this chemotherapeutic agent. And you can see on the plots on the left-hand side, you can see the cell viability as a function of the concentration in micromolar. And the experimental data are, of course, symbols. And you can see that the experiment was performed using 24 hours, 48 hours, and 72 hours incubation in both cases. And then I would like to emphasize what kind of information you can extract from this data. Of course, the information you might guess is that as you increase the concentration of the chemotherapy agent, of course, the cell viability decreases in both cases. And as you increase the incubation time from 24 hours to 48 and 72, also you can roughly see that the cell viability decreases. So these actually are the two information you can extract. And perhaps an extra information is the efficacy of one of this chemotherapeutic agent is more efficacy for the 224 cell line than it is for the EJ. So besides this perhaps three informations, it's very difficult to extract any precise information. So analysis in this case is typically qualitative, and that is what you see these days in majority of the papers published. However, I want to show you the combination of the radiofrequency therapy and one nanocomposite and the cell viability assay and a mathematical model to describe this. So here is very briefly what is the radiofrequency therapy. So in this case, you have a radiofrequency generator and you have a needle, and then you can close the circuit, thus just touching the needle in a point in the body. For instance, you can, the needle is on the tip of the electrode. You can include the needle in a particular site. You want to apply the radiofrequency, and then you choose the frequency and you choose the power. And there is a heat dissipation on the site. On the left-hand side, you can see, for instance, typical size. So typical size of the area that you can treat is something between 1 and 3 centimeter. And here is the radiofrequency electrode. So, and then you can find different areas. So the normal area around the spot, the sublethal area, and the lethal area. So this is typically, for instance, in oncology, the typical tumor size, which is around 3 centimeters to apply this radiofrequency therapy. So, and then let me show you the nanocomposite we want to combine with the radiofrequency therapy. This nanocomposite is based on graphene oxide. You can see on the left-hand side the typical chemical structure of graphene. And on top of graphene oxide, we incorporate gold nanoparticles. So gold nanoparticles about 10 nanometers in size. And also incorporate a polymer, which is the polyethylenimine. So a combination of these three materials. So, and then we end up with the nanocomposite, which is the graphene oxide, polyethylenimine, and gold. And of course, we performed all kinds of characterization of all these materials, not only characterization of the nanocomposite, but also characterization of the polymer and also the gold nanoparticles. For instance, you can see in the middle of my slide the high resolution transmission electron microscopy of the gold nanoparticles, showing the typical plane distance, which is characteristic of gold. So about 2.35 angstroms, the plane. On the right-hand side, you can see the particle size histogram of the nanocomposite. So the nanocomposite has a average size of about 27 nanometers. So let me show you the experimental design we managed to implement here. So we use actually here two different cell lines, both neoplastic cell lines, the Walker 256, also called LLCWRC 256. And we also use the second cell line, which is the B16F10. So these two cell lines were first treated only with the nanocomposite, increasing the concentration just to test the toxicity of this nanomaterial against these two cell lines. In the second experiment, we combine the application of the nanocomposite, increasing the concentration, plus the radiofrequency application. So here, radiofrequency was, the frequency was 500 kilohertz and the power around 5 watts. And so we extract the cell viability assay of all these combinations, so the two cell lines. And also we use not only the nanocomposite, but we tested also the cytotoxicity of the gold nanoparticles themselves. And then we perform a mathematical analysis of this, and the curve fitting was performed using the least square method. And how do we manage to fit the experimental data? We use actually the Hill model. So this Hill model was proposed more than 100 years ago by Archibald Vivian Hill in his second paper in 1910. So interesting to say that Archibald Vivian Hill, about 12 years after he published this paper, he won the Nobel Prize in Medicine or Physiology. And this paper was actually his second paper published. And so in that paper, Archibald Vivian Hill provides one equation to describe the cell viability as a function of the doses. So here, V is the cell viability, D is the dose, which means the concentration of a challenging molecule. And we have the Hill exponent, you can see, or Hill coefficient. K is the desorption constant of the equilibrium. And then we can extract, for instance, important parameters like the benchmark dose, BMD, and the lethal dose 50, LD50. So from this equation, we can extract the BMD, LD50. As long as you fit the data and extract the parameters like K, V0, and V1, which are scale parameters actually, and very importantly, the Hill coefficient. So here are the data. for the Walker cell line. So you can see on the upper part of my slide, the experimental data are the vertical columns, and the blue solid line is the best curve fitting of the data. So in the panels A and B actually is the cell toxicity assay for the gold nanoparticle coated with citrate. And then you see in both cases a reduction of the cell viability as a function of concentration. And on the left-hand side, panel A, is no application of radiofrequency. On the right-hand side, the radiofrequency is applied, so 500 kilohertz and 5 watts. And then you can see there is a difference in cell viability response. On the lower panel, C and D is the assay for the nanocomposite. And then you can see the increasing concentration of nanocomposite on the horizontal axis and the cell viability on the vertical axis. So again, the experimental data are the vertical columns, and solid blue line is the best fitting. And then you can see the difference. On the left-hand side, panel C, is there is no application of radiofrequency. On the right-hand side, there is application of the radiofrequency. So these are the data for the Walker cell line, and you can see about similar data for the B16F10 cell line. So on the A and B panels, the challenging material is the gold nanoparticles, surface coated with citrate, and the cell response is completely different, as you can see. So A, there is no application of radiofrequency. Panel B is the application of radiofrequency. On the two lower panels, C and D, is the application, is the use of the nanocomposite, increasing the concentration and the cell viability on the vertical axis. So on the left-hand side, the C panel is there is no application of radiofrequency. On the right-hand side, on D panel, you have the application of the radiofrequency. So once you are able to fit the experimental data like I show you on these plots using the solid blue line, so you can extract important information from this cell viability assay. And I collected this here just to show you some examples. So we have the first cell line, the Walker cell line, on the upper part of this table, and the B16F10 cell line on the lower part. So we can extract all the parameters like V0, V1, K, and E. I would like first to concentrate on the hue exponent here, or hue parameter. For the Walker cell line, you can see that the gold nanoparticle, without application of radiofrequency, we have the E value about 3. So when you apply the radiofrequency, you have a E, one order of magnitude higher, 34. For the nanocomposite, so the nanocomposite with no application of radiofrequency, you have the E exponent about 14. If you apply the radiofrequency, it drops down to 3. So it's a huge difference between application of radiofrequency and no application of radiofrequency. And that happens for both cell lines. So if you look at the B16F10 cell line, the E exponent, the hue exponent for the gold nanoparticle, without application of radiofrequency, the E value is about 3. If you apply radiofrequency, the E value goes around 1, which is 1/3. And again, with the nanocomposite, the E value is about 0.9. If you apply radiofrequency, drop down by about 1/3. However, perhaps the most interesting aspect is if you analyze the benchmark dose, or the LD50, you can see a huge difference here For instance, for the Walker cell line, so gold nanoparticles without radiofrequency, the BMD is 1.3 micrograms per ml. If you apply radiofrequency, it drops down to zero point 0.9. So you see increasing nanotoxicity. If you look at the nanocomposite, it's even more impressive. So the BMD for the nanocomposite without application of radiofrequency is 20. If you apply radiofrequency, it drops down 2 orders of magnitude, actually. It's a huge increase in cytotoxicity. So you can see the synergies between application of radiofrequency and using a nanocomposite in the site you are applying the radiofrequency. If you look at the B16F10 cell line, you can see the same pattern. So the benchmark dose for gold nanoparticles without application of radiofrequency is 0.5 micrograms per ml. If you apply the radiofrequency, it drops down 3 orders of magnitude, which is a huge difference, actually. If you look at the application using the nanocomposite, the cell line, the B16F10 cell line response is even more impressive. So the benchmark dose drops down from 119 down to 5 orders of magnitude. So you can see a huge synergies between application of radiofrequency technology and using a nanomaterial like this nanocomposite in the site. So the same trend you can look at the last column, the same trend you can find in the LD50. So the important point here is the huge synergies in combining radiofrequency therapy, which is a standard therapy, and a nano composite like I'm showing here in this talk today. And I would like to end my talk just emphasizing some points. The first point is quantitative analysis does provide extraction of important parameters. For instance, if you are using the Hue model, qualitative analysis is no way to extract any of those parameters. So quantitative analysis provides extraction of typical dose, like benchmark dose, and qualitative analysis is very difficult. Quantitative analysis provides you extraction of a typical LD50. Qualitative analysis is just rough, I would say. You just can guess, I'm sorry. And finally, quantitative analysis allows distinguished cell line bioresponse much more precisely than qualitative analysis. So once again, thank you so much for